Actually, I have a question. Because when we were talking about earlier how your group was using a negative binomial and that's kind of standard practice, but did you guys try and use some of the other ones, maybe like the mixed models or the sum?
We thought about a mixed model for a bit, but because we did have time course data, but the wrinkle was in how the data was collected. We didn't think it was like a longitudinal data set because we're working with brain tissue, and in order to get the mRNA, they had to get the brain out of the fish, and it didn't survive that. So, so we ended up deciding that we didn't really need a mixed model because we didn't think they were super correlated. And also our time points were really far apart, so we thought maybe there wasn't a lot anyway. And there was a lot more help pages for the negative binomials too. So that was a factor. But that's a good question.
So you remove the zeros or almost zero values. In this field, did they ever do like a zero greater negative binomial and then don't filter on the database? That's not done for genetics?
That's a good question, and I don't really know what the norm in the field is. From what I gather, that they do take out the zeros. I'd have to look around to see what the consensus was, but where I learned to do that is from an article in Nature, so I assumed that was something that was done a lot, but I'm not sure if other people do different things.
And one other question that I think somewhere in the code, you did the first model matrix. I think it had a no intercept. Is that?
Oh, it was 0 plus group.
So that's common to put a model out of that's what that function.
For that one, I put the 0 in there because later I wanted to do a contrast with the 3.
Okay.
So you could do it the same without, just your contrast would be different numbers. I just thought it would be more clear because I said 3 groups and then there were 3 numbers there. But definitely you could do it without it. You just have to change what your syntax later on was. Mm-hmm.
A couple slides ago, you reported a likelihood ratio test and a p-value associated with it. What's
Yeah.
the null of that test? Null Is that something new with whether the gene is expressed or to what degree it is expressed?
So the specific null for that would be in the likelihood ratio test, whatever you said to do. So if you were working with the regression model and you were saying coefficient equals 2, it would be for that parameter equals 0 in the negative binomial regression model.
Okay.
So are the genes multiple response variables, or do you have a coefficient in the model for each gene, or
So you can actually do a test for each gene. So there is a sequence of counts, and let me go back to the data. Yeah. So there's one gene and then a sequence of responses based on however many samples you have. And I imagine you could have this go farther and you'd have more than 6 or 7 that I put on the slide. So the test is for just one gene.
whether it's different if
Mm-hmm. Yeah.
You talked a little bit about how certain packages were faster than other ones. How much is that a constraint? How much basic does it take to
So how big are the data sets, and is it important for it to be fast?
Could you run into speed issues?
We did run into size issues. The speed issue is really how fast you need your results. So I think we estimated, because we had, 40 million reads per sample, and then we had 64 samples. So we estimated we could get it done in half a day or something by using the cluster. But if you didn't have a cluster, or if you're already kind of straining on the data set, or maybe if you had like a lot more data than we did, then maybe that could be an issue. Because these data sets can be big.
I guess the follow to this question. So this is sort of like the data set that goes into the model, so our observations are genes. Or for those of our observations are genes. Is that the right way to think about it? Or
So like you're doing a test for the genes, so like each one. So like your like samples would be like your observation. Like these are like, all the Y's in like one row.
In the multivariate data, the sample data of a unit. One sample is the same unit data set, and there's data for each gene in for each unit.
So you're saying, can you phrase that again?
One's the sample unit of the gene, or the sample mashed together brain cell.
So you're looking at each gene separately, yeah. So like you're doing one test for each gene, which is why I was talking about the multiple comparison thing.
So when you fit the model, it's actually fitting a model for each separate gene, and then you're just putting all those key values and then you're ordering them one by one.
Yeah.
in the columns here, different fish rays or different parts.
Yeah, so like, I took this from a paper, so I'd have to look up what the CT and PA mean, but it would be like, maybe one would be treatment C, and then it was on line PA or something, like,
But I can think of the columns here, like different people with
Yeah.
okay.
mhm.